Alright, this is Jahangir from MedPorium, and today we have part 6 of our Genetics and Cellular Function unit, in which we will discuss chromosomes and heredity. To start, we need to understand what chromosomes are. Chromosomes are defined as a structure that carries genetic information and is composed of DNA wrapped around histones in a thin fiber known as chromatin, which is housed in the nucleus of the cell. Now let's define heredity. Now heredity is defined as a transmission of genetic characteristics from parent to offspring, and these genetic characteristics are what give us our physical traits, from the color of our hair to our blood type. To understand our 46 chromosomes, we need to discuss the karyotype. Now the karyotype gives us a visual understanding of our 23 paired chromosomes in numerical order. Here's a picture of what the karyotype may look like. As you can see, we have 2 chromosomes in each pair, which are known as homologous chromosomes since they have an identical length, centromere position, and gene position, which is known as a locus. Now I'm sure you're wondering, but Jahangir, I only see 1 strand in each pair instead of the regular X structure that I usually see for the chromosomes. Well, the karyotype chart shows chromosomes in metaphase, in which both sister chromatides are in contact. The chromosomes may look like a single strand due to the close contact these sister chromatides experience. This is what they look like zoomed in. Now, one thing to remember is that each chromosome in the 23 pairs are from your mother or from your father. For example, this chromosome would be from your mother, while this chromosome would be from your father. The same is seen in the sex chromosomes, which are known as the X and Y chromosomes. Now, the sex chromosomes are much different than the rest of the chromosomes in that they determine the sex of the offspring. The mother can only supply the X chromosome, while the father can supply both the X and Y chromosomes. So all in all, the father determines the gender. Females have a double X chromosome pair, while males have an X and Y pair. Since the X chromosome differs from the Y chromosome, this pair is known as a non-homologous chromosome, since their height, location of the centromere, gene position contrast one another. Now, one thing to note is that the Y chromosome does not have a functional purpose other than it gives the gender of a male. Another thing to note is that all chromosomes besides the sex chromosomes are known as autosomes. A cell with 23 pairs of chromosomes are known as diploid cells, and a cell with 23 unpaired chromosomes are known as haploid cells. So in all, haploid cells have 1 chromosome for each of the 23 pairs of chromosomes that are in the karyotype. An example of a diploid cell would be almost every cell in your body. An example of haploid cells would be the sperm and egg cells. One more thing before we move on. The belief that we only have 46 chromosomes in total throughout our body is a myth. We do have 46 chromosomes, but these 46 are housed in nearly every cell's nucleus of the diploid cells. Essentially, we have 46 different types of chromosomes that account for physical or mental traits. Now let's talk about genes and alleles. As we discussed in the first part, genes are a segment of DNA that code for a specific protein. An allele is a contrasted version of a gene that can appear due to mutation. This is what an allele may look like. Although a homologous chromosome has the same locus, which is the location of a particular gene in the chromosome, and the same gene, they have different versions of that gene, which is known as our allele. There are 2 different types of alleles. One is dominant and one is recessive. Normally, dominant alleles code for normal functional protein and are usually represented by an uppercase letter. For example, the uppercase D, as you can see right here, would be our dominant allele. Now, recessive alleles code for a non-functional variant of protein. They're usually shown as a lowercase letter. The lowercase letter d, as you can see above, is an example of a recessive allele. With those 2 alleles, they can either be homozygous or heterozygous when they are paired together. A homozygous pair indicates 2 identical alleles, while a heterozygous pair indicates 2 different opposing alleles. Remember, 2 alleles usually code for a gene. For example, the heterozygous pair dominant D and recessive D codes for, let's just say, brown eyes. We inherit these 2 alleles from our mother and our father. So we can say that we inherited the dominant allele for brown eyes from our mother and the recessive allele for blue eyes from our father. So the dominant allele would be that capital D, the uppercase D, and the recessive allele would be that lowercase d. Now, determining the likelihood of an offspring obtaining a specific genetic trait from their parents is shown through the Punnet Square. So here's what our Punnet Square may look like. Today we will determine the genetic likelihood of sickle cell anemia for an offspring whose mother and father are both carriers of the gene. This means the mother and the father have a heterozygous pair. So in this description, hemoglobin A or HbA indicates the normal gene for normal adult hemoglobin, and hemoglobin S or HbS indicates the gene for sickle cell hemoglobin. In this Punnet square, hemoglobin A will be the dominant allele, while hemoglobin S will be the recessive allele. Now the superior portion of the Punnet square will be the mother's heterozygous genotype, while the lateral portion of the Punnet square will be the father's heterozygous genotype. We'll talk about genotypes later. The boxes in the Punnet square represent the offspring. Now, as you can see, the mother and the father have a heterozygous genotype, meaning they both have one allele for hemoglobin A and one allele for hemoglobin S. Now, you might be wondering, what is a genotype? Well, a genotype is a pair of alleles one possesses for a specific unit. For example, the allele pair hemoglobin A and hemoglobin S is a genotype. Now let's determine the percent likelihood for their offspring to have sickle cell anemia. Remember, the dominant allele will demonstrate their gene over the recessive allele, so the dominant allele will always dominate over the recessive allele. Now what does this mean? Well, when we have a heterozygous pair, our dominant allele will demonstrate its phenotype before the recessive allele. Now you might be wondering, what is a phenotype? Well, a phenotype is much more different than a genotype in that it is an observant trait instead of a pair of alleles. For example, hemoglobin A and hemoglobin S would be the genotype, but a normal adult hemoglobin would be the phenotype. So the phenotype is what is shown as a physical trait. Now let's solve this Punnet square. Alright, so we've finished filling the Punnet square. Now what? Now we determine the percent of offspring who have the sickle cell trait, meaning they have a heterozygous genotype, the percent of offspring who have the sickle cell anemia, or those who have the hemoglobin S homozygous genotype, and the percent of offspring who will have the normal adult hemoglobin, or those who have the hemoglobin A homozygous genotype. As you can see, 50% of our offspring will have the sickle cell trait in terms of 4 offspring in total. 25% of our offspring will have sickle cell anemia, and another 25% of our offspring will have normal adult hemoglobin. This means 75% of our total offspring, and our total offspring is 4, will have the hemoglobin S allele, while 25% of our total offspring will not have the hemoglobin S allele. Punnet square is a great resource in determining the genetic likelihood for an offspring to have a certain gene. Alright, and unfortunately that wraps up part 6 of our genetics and cellular function unit, in which we discussed chromosomes and heredity, including genes and alleles. And as always, a Pomodoro day will make you a doctor someday. I'll see you next time.